Thank you, Sam, for the introduction. I would like to thank ISHR for giving me this opportunity to present my work today, where I'll be discussing about branched-chain amino acid catabolism in the heart. So branched-chain amino acids, or BCAs, are comprised of the three essential amino acids, leucine, isoleucine, and valine, and they have been attributed with diverse physiological roles like maintaining the balance between protein synthesis and breakdown, and modulating glucose and fatty acid metabolism in various tissues. Now, the BCAs can enter into circulation when they are released from the proteins, either from diet or from other tissues. Alternatively, they can be cleared from circulation when they are reincorporated into new proteins, while the remaining BCAs in circulation can be oxidized in various other tissues. Although BCAs have beneficial effects under negative energy conditions, chronic increases in circulating BCAs have emerged as a biomarker for several obesity-associated metabolic diseases over the past decade. Indeed, increased plasma BCA levels have been associated with obesity and insulin resistance in humans, Zucker fatty rats, diet-induced obese rats, as well as leptin-deficient OB mice. This has led to the hypothesis that catabolic defects of the BCA pathway or impaired metabolism of BCAs could result in increased accumulation of the circulating BCAs observed in metabolic disorders like type 2 diabetes and associated comorbidities like cardiovascular diseases. To give a brief overview of the BCA catabolic pathway, the BCAs can be reversibly transaminated to form branched-chain keto acids or BCKAs by the enzyme branched-chain aminotransferase or BCAT. The fate of the BCKAs then depend on the activity status of this enzyme supercomplex called branched-chain keto acid dehydrogenase or BCKDH. BCKDH can be inactivated by phosphorylation by this kinase called BCKDK, and that does not allow the oxidation of the branched-chain keto acids. On the other hand, the phosphatase PPM1K can dephosphorylate BCKDH complex and activate it, allowing the BCKA oxidation by various distal BCA catabolic enzymes, ultimately resulting in the formation of the three main acyl-CoA species, acetyl-CoA, acetoacetyl-CoA, and methyl-malonyl-CoA, which can in turn act as intermediates in various metabolic pathways. Now, several studies have now identified various pathological stress signals that can impair the BCA catabolism, which might eventually lead to increased circulating BCA levels. We found that in mice fed high-fat, high-sucrose diet for 16 weeks, there was a significant reduction in both the proximal as well as distal BCA catabolic genes within the heart. Further, high-fat, high-sucrose diet also resulted in a significant reduction in KLF15 levels in the obese heart, and KLF15 is an important transcription factor for this catabolic pathway. This reduced catabolic gene expression also resulted in increased serum branched-chain keto acid levels, suggesting that the BCA catabolic gene expression in the heart can be suppressed in response to an obesogenic milieu. Now, our lab is part of a OPOS clinical trial involving patients with obesity that are enrolled at the St. John Regional Hospital to undergo elective cardiac surgery. So we could procure the atrial appendage and the subcutaneous adipose tissue surrounding the heart from these patients during surgery. And for the purpose of this study, we stratified these patients into different classes of obesity based on their BMI. We found that in the subcutaneous adipose tissue, the branched-chain keto acid dehydrogenase enzyme activity was significantly downregulated in the obese patients, and consistently we observed increased protein expression of the kinase BCKDK and a corresponding increase in the inactive phosphorylated BCKDH. The transcription factor KLF15 was also significantly reduced in the obese adipose tissue. Further, we found that this decreased BCA catabolism within the adipose tissue was positively correlated with increased length of stay in the hospital following surgery in these patients, suggesting that the impaired BCA catabolic gene expression within the adipose can influence the post-operative outcomes. We next investigated what happens to BCA catabolism within the atrial appendage. and found that there was significant increase in the BCA levels within the atrial appendage in the obese patients. However, we did not find any reduction in BCKDH activity, which could also explain the reduced keto acid to amino acid ratio that we observed in the atrial appendage of the obese patients. Further, this reduced BCKA to BCA ratio was significantly correlated with increased BMI as well as length of stay in the hospital following surgery. So together we theorize that the reduced BCA catabolism within the subcutaneous adipose tissue might result in shunting of excess BCAs towards the heart for oxidation. However, the excess BCA entry into the heart cannot be effectively catabolized by the enzymes within the atrial appendage, and that could contribute to the adverse outcomes. So our data is in agreement with previous studies that have demonstrated impaired cardiac function in mouse models of BCA catabolic defects, and that those studies have also reported increases in the branched-chain keto acid levels. Further, different studies have now demonstrated the direct effect of the BCKAs in impairing mitochondrial respiration, compromising glucose uptake, and increasing oxidative stress within the cardiomyocytes. We therefore hypothesize that at least some of these metabolic complications mediated by the branched-chain keto acids could be explained by its effect on cardiac insulin signaling. So we examined insulin signaling in response to acute exposure of individual keto acids in cardiomyocytes isolated from neonatal or adult rats. Pre-incubating with the keto acids significantly reduced insulin-induced AKT phosphorylation as well as impaired AKT activity, suggesting that exogenous BCKAs can impair insulin signaling within the cardiomyocytes. We next investigated whether altering endogenous BCKA levels by genetic modulation of the BCKDH complex could influence insulin signaling. For this, we targeted the BCKDH-A subunit of the BCKDH supercomplex, and we found that silencing BCKDH-A in the neonatal rat cardiomyocytes significantly increased the intracellular BCKA accumulation, and that resulted in impaired insulin-induced AKT phosphorylation, as observed previously with the exogenous BCKA treatment. Conversely, overexpressing BCKDH-A resulted in a modest but significant reduction in the intracellular BCKA levels, and that could potentiate the insulin-induced AKT phosphorylation in the cardiomyocytes. Together, our data suggests that affecting the clearance of the branched-chain keto acids by genetic modulation of BCKDH-A can reciprocally regulate insulin signaling in cardiomyocytes. So to summarize our findings, we found that the BCA catabolism in the subcutaneous adipose tissue and atrial appendage is altered in obese patients undergoing cardiac surgery, and this altered BCA catabolism could correlate with adverse post-operative outcomes. We also show that in diet-induced obese mice, the BCA catabolic enzymes within the heart is suppressed, and that results in increased circulating BCKA levels. Further, we show that an acute exposure of individual BCKAs can directly inhibit insulin-induced AKT phosphorylation in cardiomyocytes. And finally, altering the intracellular BCKA levels by either overexpressing or silencing BCKDHA can reciprocally regulate insulin signaling within the cardiomyocytes. So I would like to end my talk with acknowledging my supervisor, Dr. Thomas Pulinen-Kunnel, Dr. Kinnisberger, and all our lab members, the IMPART team for the clinical samples, and the funding agencies. Thank you.
So the first question is for Dipsikha from Gary Loposchak. Nice talk. How do you think BCKA are modifying phosphorylation of AKT? And then the follow-up question, are they interacting directly with AKT?
Thank you, Gary, for the question. We did see that BCKAs could directly activate mTOR signaling. So we think that that is one of the way that it is modifying AKT phosphorylation. And also we found that BCKAs can inhibit insulin-induced PP2A inhibition. So that is also another method that we feel. And as it is known, and we have also found that BCKAs can directly impact mitochondrial respiration and increase ROS, so which can also influence AKT phosphorylation. So right now, actually, we have a couple of theories surrounding this.
The next question we have is from an anonymous attendee. They're talking about the fibroblast cell cultures. They ask, is cluster 8 a non-fibroblast cell culture the most deviating one, is what they mention. So I believe this question is for Alexandra Garvin.
Yes. Thank you for that question. We were worried about the same thing when we first saw the clusters. However, none of our cells express markers of either endothelial cells or smooth muscle cells or even hematopoietic cells. And so we know that these are fibroblasts. However, they are just a very unique subset, as is cluster 7. And one theory we have is that potentially these fibroblasts may potentially expand in different either injury states or other ways, but we're certain that those are fibroblasts. Why they're so unique, we're not incredibly sure.
Okay, so the next question is from Julie. It's a technical question for Dipsikha. How did you measure BCAA level, whether you use a mass spectroscopy?
Yes, we indeed used UPLC MSMS for the intracellular as well as the serum and plasma BCA and BCKA levels. Yeah.
Then we're going to jump back to Alexandra. The next question is, did you do spatial selection of the fibroblasts you subjected to RNA sequencing?
I'm sorry, I'm not exactly sure what you mean by spatial selection. If you mean localization to the heart, we just did a complete LV digestion and isolated all fibroblasts from the left ventricle. I'm sorry if that doesn't answer your question, but.
Okay, so the next question is also for you, Alessandra, from Sina. Is it possible to distinguish which portion of the heart each cluster of fibroblast comes from, that is closer to the epicardium versus closer to the endocardium?
As of right now, our analysis does not distinguish that, but some follow-up studies that we will be doing is some imaging in the heart and staining with some markers of these clusters in the left ventricle to see if potentially there are special localization for some of these clusters. And I'm very interested in that, actually, to know if some of these phenotypes are coming from different areas of the heart. So hopefully that is to come, but right now we don't know.
The next question is also for you, Alexandra. They ask, did you compare these fibroblast clusters to a non-hypertensive rat ventricle fibroblasts? And if so, which clusters are specific for hypertension?
We did not compare these to non-hypertensive hearts. The main question for the study was within the hypertensive heart, what the impact of ACE inhibition was. And so we did not have that non-hypertensive group in this study, and we haven't done that.
Okay, so the next question is also for Alessandra from Leslie Kennedy. So I think it's similar question what you just answered, but still I will just read out. Is there any evidence of fibroblast in differing region of ventricular myocardia having distinct gene expression? In other words, do you think the cluster group in cardiac region, or do you think the clusters may be more heterogeneous, spread throughout the myocardium? This may impact crosstalk between fibroblasts and cardiomyocytes in the left ventricle versus those are in the right ventricle.
Yes, great question. And I agree with you that potentially different localization of these clusters would impact the crosstalk. And although right now we don't know, I wouldn't be surprised if we find some of these clusters are in particular areas. I know that there's some evidence out there to suggest that fibroblasts from either the endo or the epicardium serve slightly different roles, and I'm eager to know the question to this, or the answer to this question.
Great. The next question is, how do you isolate the LV fibroblasts for single cell RNA sequencing?
Sure. So after we isolate the left ventricle, we mince up the tissue and put it through a couple rounds of digestion with an enzyme called liberase. And the fibroblasts that are released from that digestion are then plated, and since fibroblasts are so adherent, we use those adherent collagen producing cells, which is what we used for this experiment.
Alexandra, may I just interject? So maybe they mean, which methodology do you use to isolate cells? Do you use Langendorf, non-Langendorf methodology?
Oh, so we don't cannulate the hearts, and we don't digest the heart when it's in its full form. I know some people will perfuse an enzyme, but what we do is we just mince up the tissue, after it's removed, and then from that minced up tissue, we submerge it in the liberase buffer and put it through several digestions and collect the supernatant, and then let the cells adhere, if that clarifies.
Okay, so next question is from Ron. He's asking two questions. Were the RNA sequence experiments from the hearts at a time point with overt tissue fibrosis? And the next question is, any plans to build a progressive data set over the progression from hypertension to HT to dysfunction?
Yes. So at this point, the hearts are certainly hypertrophied, and there is an amount of fibrosis, but it's not overt. I would not call it overt fibrosis. If you follow up with other injurious stimuli, you can stimulate higher amounts of fibrosis in these hearts at this time. And as for the second question, any plans to build a progressive data set? We would love to with RNA-seq, and it is, I think, an overall goal to understand the timeline of these changes, but right now we're focusing on how responsive this cell type is to our treatment that you'll hear about on Friday.
Oh, sorry, I'm talking when I'm muted. The next question's for you, Alexandra. From Mohan, they ask, could you please elaborate on the enzyme cocktail used for single cell preparation?
I think I already answered that. If you mean for cell isolation, we use liberase as our enzyme.
Okay, so the next question is also for you, Alessandra. This is from Jun Sun. He's asking, besides co-treatment with ACE inhibitor enalapril in angiotensin II induced hypertension, have you tried to treat the rats with nitric oxide donor? If so, is there any difference in cell cluster between angiotensin II plus enalapril and angiotensin II plus NO donor?
So good question. We haven't done those exact experiments, but previous experiments in our lab have utilized a NOS inhibitor. So not a donor, but in our hypertensive hearts that either got the transient ACE inhibition or not. They also then followed up with an L-name NOS inhibitor. And in those studies, isolating the cardiac fibroblasts and then looking at them in vitro, our transient treatment does protect against some of the impacts of that L-name treatment in terms of proliferation and collagen gene expression. So we know that this transient treatment does play a role in protecting against some of those pathways, but in terms of the single cell sequencing, we haven't done those experiments.
All right, the next question, and maybe if individuals would like further or additional information, they can feel free to reach out to the individual speakers. if they feel like the answers weren't adequate to kind of what they're asking for. But the next question to Alexandra as well is, while classifying the 9 subpopulations into 3 classes, what were the criteria being used to group them into 3? For instance, does it mean the subpopulations of fibroblasts classified under oxidative stress are programmed to express different kinds of oxidative stress response genes?
So the way that I went about, going through this data is we have thousands of different differentially expressed genes, and what we were looking for were, I chose these 3 categories because of the roles that they play in fibroblast activation. And so in terms of actually grouping a cluster into one of those areas, that's a little bit harder to do, but more just looking at overall, was this cluster more proliferative or more inflammatory or more fibrotic, was a way for us to help understand potential contributions of these clusters to overall, cardiac fibrosis. And so I wouldn't say that we've necessarily pinned them down to be one or the other, but just over all of the genes that were, uniquely expressed in a cluster, we pulled out these processes because they were relevant to our questions and because they were areas where we saw some unique characteristics between the clusters, just to help understand their contributions.
So Alexandra, Richard is wondering, what are the markers did you use to identify your fibroblast population after isolation?
Markers in terms of purity, I think, is what they're asking. So ultimately, we know that all of the cells that we isolated express things like vimentin, and that they don't express markers of other cell types that we may potentially get in this prep, such as vascular smooth muscle cells or endothelial cells or immune cells. We didn't see CD68 or CD31, smooth-lin, any of those markers of potential contaminating cells in a fibroblast prep. So we're confident that we're looking at mainly, collagen-producing fibroblast cells.
All right, the next question from George is, do you know if the change in fibroblast cluster morphology in response to hypertension is terminally differentiated or reversible? And then there's a second question. Well, I'll read them both, then you can answer them both. And the second question is, do you know whether female mice undergo the same transformation in fibroblast differentiation in respect to insulin, given the sex-specific differences in collagen deposition?
Okay, great question. So the first one, we don't know in terms of, differentiation, whether these cell, states may be reversed either with a treatment or, if they are reversible at all, or if potentially these cells might, die out and be repopulated. We don't know if these are reversible phenotypes. But all interesting questions in how, a targeted treatment may impact these clusters. And then the second question, we have not done any studies with females, but of course that would be in the future directions, and knowing how different they are and how relatively protected a female heart is, it would be interesting to see if these same clusters would come up.
Okay, so now Monica is asking, how I missed it. Okay, so pure, how you isolate pure population of cardiac fibroblast from the heart. Then the second part of her question is also, it seems that you have 4 distinct population of fibroblast. Wouldn't you expect to have more visible transition from one population to another?
So I think hopefully you've heard my answer to how we isolate the fibroblasts. So I'll, in the interest of time, move on to the second part. So we get 9 distinct clusters. I focused in on 4 clusters today, given their unique characteristics in areas of proliferation and fibrosis and inflammation. And so whether I would expect to see more visible transition from one population to another, If you're referring to the UMAP plot, where it's basically a cloud of cells with varying colors, I think that has to do with the fact that we're using a very pure population here. They were, rested in, culture dishes for 3 days. So they were adherent. They were growing for 3 days. This is a very pure population. So we don't get huge changes in gene expression, like you might even see with different cell types in the heart when people isolate all the cells from a heart. So because these are, cardiac fibroblasts that were cultured for 3 days and then subjected to single cell RNA-seq, the fact that we even saw distinctions was exciting. And even more so the fact that we see an impact of our treatment, again, which you'll hear more about on Friday, was even more exciting. So I can't say that we would have expected to see a more visible transition given the fact that we were working with such a purified population of cells.
Great, thank you. We're going to switch over to Natasha now. Natasha, the question for you is, would C144S alter the ubiquitin ligase activity of TRIM72?
Okay, thanks actually for that question. So that was one of the first things that I actually looked at in hearts from these mice, because its ubiquitin ligase activity is believed to affect IRS1, cause its degradation. So I tried to look at overall ubiquitination in the hearts, and I didn't see a change in that, and I didn't see a change in targets of TRIM72 that are expected to be changed if its activity was changed. Having said that, I did not specifically run an assay looking at TRIM72's ubiquitin ligase activity. So if that answers the question.
So the next attendees is asking or wondering whether the effect you see is the enzymatic dependent protection or structural.
So sorry, I'm not specifically sure what that means, but what I'll say is I didn't see evidence of enzymatically affecting its enzymatic activity in terms of ubiquitin ligase activity. But other than that, I don't know the answer to that question.
All right, we're going to switch back over to Alexandria. And I believe you've already asked or answered this question, but just to make sure it's adequately addressed. The question is, did you find any differences in the cluster depending on the location in the heart?
Right, and unfortunately we don't know. We isolated fibroblasts from the heart altogether, and so unfortunately we don't know where each of these clusters are coming from, if they are specifically located, but something we plan to follow up on with some candidate markers using some imaging of the left ventricle.
Let's go back to our first speaker, Dipsikha. So Azrul is asking, what is the glycogen content? The second question is, does it affect Krebs cycle intermediates?
Okay, thank you for this question. We did not measure the glycogen content, but there has been studies, a recent study actually in a PPM1K phosphatase knockout mice, where they did find differences in glycogen metabolic intermediates, and they also incubated HepG2 cells, liver cells, with BCKAs, and they found an induction in glycogen phosphorylase mRNA level, which is responsible for glycogen breakdown. But it will be interesting to see what happens within the cardiomyocytes. For the second question, we are in the process of developing a method to measure the TCA cycle intermediates, but then again, previous studies have shown that if the BCK oxidation is inhibited, that results in reduced TCA cycle intermediate. So we are expecting a similar result with our study as well.
Thank you. All right, we're going to come back to Natasha. Was there a sex difference in the ischemia reperfusion results, and are females also cardioprotected?
So we didn't specifically compare male versus female TRIM72 knock-in mice in in the ischemia reperfusion experiments. But having said that, we had a couple of female mice that were done, and they also showed some protection compared to the wild type mice.
So the next question is from Emma, and it's for Alessandra. I think the Srinivas part is you already answered, so I'm directly going to the second part of her question is, have you compared your single cell data and cluster with other model, such as the more ischemic model by Gladka et al. 2018?
Yeah, thanks for that question. And I know that paper you are speaking of, and if I recall, they looked in all cell types of the heart, cardiomyocytes and fibroblasts, and that they did find very distinct clusters. They found fewer clusters than we did, but very distinct ones. And what I have found in general is when comparing the clusters that we have to those from models of ischemia, post-MI, that those clusters tend to have much more extreme expression of markers such as periostin and these other markers that are really highly upregulated after an injury that we're not necessarily seeing differentially expressed. They're expressed in our cells, but not very highly, and not in distinctive clusters. And so in terms of comparing our data sets, I think it's relatively difficult given the methodological differences, but also I think ischemic hearts might show more extreme expression of some of those markers that we're not necessarily getting in our hypertrophied heart. I hope that answers your question.
The next question's from Svem and Alexandria. They're wondering if you compared your hypertrophy data set with the single cell data from mice after MI published by Richard Harvey's lab. And is there a specific cluster in both data sets contributing to cardiac fibrosis and scarring?
So thanks for that. And I think my answer will be similar to my previous one in that I can't say I've compared it to the specific data set, but to MI data sets in general, those data sets tend to have more extreme expression of activated cell markers. But whether there's a specific cluster in the data sets contributing to cardiac fibrosis, I can't speak for the other data set, but for ours, our cluster 1 really tends to express genes that characterize, an activated phenotype. And so in our data set, we're really zeroing in on really one cluster being the most activated and potentially most pathogenic. And so it'll be interesting to see how, our further verification of these clusters and potential localization in the heart, how this might impact overall cardiac remodeling.
Okay, so a couple of technical questions for you, Alessandra. So when fibroblasts were plated, what substrate did you use? And the second question is, and how long were the fibroblasts were plated?
Yes, so we plated on plastic, and they were plated for 3 days. And we are well aware of the activating, that plastic can activate the fibroblasts. And so knowing this, as we look at our data set, it's almost interesting to see, especially with the impact of our treatment, the fact that, so we recognize this as a potential limitation, but also the fact that plastic can potentially activate our cells. It may also help pull out the differences between our treated and control cells as well. So yes, we plated them for 3 days. They did not grow to complete confluence, but they were plated on plastic just to slightly expand the population for the sequencing.
Thank you. We're going to jump back over to Dipsikha. What effect does BCAA have on the respiration capacity of the cardiomyocytes?
So we measured the effect of BCKAs on the respiration capacity by seahorse, and we did find that it suppresses the basal as well as FCCP-induced maximal respiration rates. And it also resulted in reduced ATP production within the cardiomyocytes.
Okay, so the next question is for me. So thank you, June. This is ECI ISHR banner, but no, it's not from the big TV screen. Due to intellectual property, I cannot disclose the exact mechanism. But jokes apart, next question is for Natasha actually. And so the question is TRIM72 specifically expressed in cardiomyocyte or other heart cell as well? How do you think, that second part of the question is, how do you think the nitrosylation at cysteine 144 affect the other cells such as fibroblast? Third question, does cysteine 144 nitrosylation affect ROS generation?
Okay, so first, TRIM72 has been reported by some recent papers to affect fibroblasts, so it seems to be expressed in cardiac fibroblasts. I don't know how nitrosylation of cysteine 144 would specifically affect fibroblasts. What I can say is that there was a cell culture experiment that was published in Mark Kor's paper a few years ago before this study was done. And it also showed that this specific type of mutation, where you get cysteine 144 mutated to serine, protected against hydrogen peroxide induced cell death. And so, I mean, I would expect that as nitrosylation would also protect against other types of cell death. And then as far as the effect on ROS generation, I don't know. I didn't look at that, but I would expect that by protecting against further injury, you would keep there from being as much ROS generation, but I didn't test that. Okay.
Great. And the last question for you, Natasha, from Jader is the specific mutation in TRIM72 affecting mitochondrial function during ischemia reperfusion injury?
Yeah, I would love to know the answer to that question, but I did not look at that during ischemia reperfusion injury. And again, I would kind of expect that by protecting against further injury, the mitochondria would be overall healthier compared to the wild type mice, but I don't know that that would be a specific function of the TRIM72 mutation versus a byproduct of the protection.
Thank you, everyone. We've reached the end of our questions, and so I think we'll go ahead and wrap up at this time. We just want to thank one last time the three presenters that we had today. Thank you so much for participating in this webinar and for sharing your science with us. We really appreciate you being here today. And one more plug for the other webinars that we have ongoing this week. Please check them out. We have some great science that's going on here. So thank you for joining us today.
Thank you.
Thank you. I mean, if you don't mind, I would just like to personally thank the three of you. Dipsikha, Alexandra, Natasha, great stuff, great webinars. You were a bit of a kind of, to some extent, guinea pigs, because this was the ECI-MCI first webinar, and I think it went really well. We had great numbers of attendees, and I think the Q&A session was fantastic. And last, I'd like to thank Sam and Sarah. Great sharing. I mean, they've constructed this, by the way, this ECI-MCI session, and I'd like to thank them because I think this is really needed, as on top of, obviously, you know, the other webinars that we've had. And we've got 2 more coming up as well. Is that right?
Yes,
Yeah.
there are 2 more. Yeah.
Yep.
So please come and join us, everyone. So thank you once again.
Thanks, guys. Have a wonderful day.